Okay, so my name is Michael Themis. You must be getting tired by now, listening to all this. I'm going to try and explain it in the most easiest possible way, so it's not too scientific. And it's a very interesting concept because the title of my talk is Fetal Gene Therapy, right? Treating children before birth, and underneath is very important. Are you serious? Should we be doing this? Think about it ethically. The person doesn't get a choice. Lots of things to think about. Now, gene therapy, we have to say, what is it? What is gene therapy? We know there are lots of genetic diseases. We know that genes are usually the cause of these genetic diseases. And so if you want to treat patients with drugs, you can carry on doing it. If an individual has haemophilia, they can't clot their blood, they're missing a protein called factor IX. And there's a story about a child in a playground falls down, starts bleeding because they cut his knee, has to go to hospital 3 weeks later, £40,000 later. Lots of this protein are given to this child, factor IX, to cure the patient. Child goes back to school, falls over again next week. It's a really difficult situation, expensive for the NHS. Now, what if you could take the gene that makes that factor IX and you could put it into that person just once and they're cured for the rest of their lives? Wouldn't that be an easy route? And that's the concept of gene therapy, putting a gene into somebody that has a disease because their gene's faulty. It's as simple as that. So, because of all this technology, and because the Human Genome Project has identified lots of different genes which have caused genetic diseases, gene therapy was born. And without going into too much detail, there's many diseases have been treated now by gene therapy. It's been done worldwide, mainly in the US. There's all sorts of ways of putting the genes in, some better than others, all sorts of different diseases, as I said. So there's been over 3,000 clinical trials to date. Some of the big players are getting involved in it. GSK, Novartis, they're making these products in order to give the genes. And the hardest thing is that's taken us such a long time is to say, how can we get a gene into a person, into all the cells that you need to treat? It's very difficult. It's taken us such a long time because we didn't know not only enough about the biology, but how to get hold of the gene and then put it in. You could say it's impossible. And I start my lectures to students to say, first of all, I'm going to tell you it's not possible. And then for the rest of the time, me giving you these lectures over the next 10 weeks, I've got to persuade you we can do it. And so I'm going to try and do that a little bit to you now. So the investment in gene therapy is huge. Billions at the moment are being pumped into gene therapy. So gene therapy, you may have heard of it on the news, really exciting technology, a one-off delivery of a gene to a person that needs it. That's what gene therapy is all about. So I do gene therapy, as you may have guessed. Now, how do we get genes into patient cells? Well, for years, viruses have infected us because they've evolved very clever ways of getting into our cells and taking over and doing us damage. And so what we do is we hijack them. We take viruses, we take them apart, we take the bad bits out, we put our gene in them, and we use those viruses to infect people. That's how gene therapy delivers. Now, earlier on, we couldn't do that at all, so that's why it's impossible. But now we hijack the viruses, one example being HIV. All those years, you probably, some of you, well, you're all younger than me probably, but some of you remember all this big deal about AIDS and HIV, and we learned all the money that went in from the government to teach us how to take HIV and understand the biology. Now what we've done is taken HIV, taken it apart, put genes in it, and use it to deliver our genes for gene therapy. So there are a number of viruses. Adenoviruses, we've all been infected before by adenoviruses. They give us a sort of cold symptoms. AAV is a new type of virus which they found contaminating adenovirus, but actually found that they're actually quite good at getting into cells. So that's a nice one. Retroviruses, well, HIV is a type of retrovirus. So we've got that. And then you can just put DNA on its own, but if I was to put DNA into any person, it would usually just be destroyed by the blood in the blood, and it doesn't work very well. So we have a choice. We have a menu here of ways of getting genes into people. I was going to go about this life cycle, but essentially what they do, viruses infect cells, they go into the nucleus of a cell, and they deliver the gene, and then bingo, off we go. We cure someone. Now, this poor little individual at the top here, this young man suffered from severe combined immunodeficiency. In other words, had no immunity. He lived 12 years of his life in a bubble, a plastic, sterile bubble, bubble boy's disease, they called it. That's just the most awful thing. This is the thing that drives us every day to try and do some research to try and help these people. Now, this individual, he suffered, but what scientists found is they could take stem cells from these patients, they could put the gene into the stem cells, and then put the stem cells back into that patient, and then suddenly they've got an immunity, because they've treated the cells, it's the patient's own cells, so the patient doesn't sort of reject them, and suddenly people were being cured. Now, I don't expect you to you can read this list of different diseases, it's all a bit complex. X-skid is the one I explained to you, but then there's you've heard of beta thalassemia, you've heard of haemophilia. People with cancer could be treated now with cancer gene therapy. That I'll tell you about in a minute. People have been made to see again when they were blind, and there are products on the market. So this is really going somewhere. This is really exciting. And as I said, the investment is huge. Now, the trouble is, you see a little asterisk on some of those, because when we got all excited and we were flying into the clinic and we did all this work, after treating some of these people, these children, they got leukemia. And the reason they got leukemia is because the viruses that we were using to deliver the genes were damaging cells, damaging genes inside the genome of these patients and activating cancer genes. So these patients, some of them got cancers from the gene therapy. So then you could argue, well, is it safe? Should we do it? So then I embarked on a program at the time there. I thought, we know there are problems, gene therapy, trying to reach in adults, trying to reach some of the diseases manifest before birth in the fetus. Wouldn't it be nice then to find, to avoid that? It's difficult in an adult to deliver a gene and get all the, say you were trying to do the whole, deliver to the liver or all the muscles in the body. It's difficult to reach all those cells with all your viruses. They don't all get infected, so you can't all get a gene delivered to them. And we have an immunity. So if you put something foreign in our bodies, whether it be a virus or something, we're going to attack it and get rid of it. So these are all problems for treating adults in gene therapy. So the idea was, what about before the disease manifests itself, we could deliver the gene in utero. In utero, there's no developed immune system. That's a good thing for us to do gene therapy. We could treat before the disease causes a problem. We might reach very early types of cells, the stem cells, to treat them early on. And there's a whole load of reasons why I could justify going in before birth. So you might say, you're talking about going in, treating an individual before they're born. They don't get a choice here, but they might be born totally cured and have a perfectly good life. So I could argue both ways. And if any of you have an opinion on that, please stop me and say, well, this is what I think. Because at the end of the day, you might think it's a bit crazy to treat an unborn. Or you might say, what would you choose? You would treat your unborn if you think we could cure that person. So what I've got to prove to you, if I'm a scientist and I expect to go to the clinic with this idea, I expect to say to you, or prove to you, it's safe. Because if it's not safe, obviously we shouldn't do it. So we did some work where obviously you can't just sort of go to someone at the bus stop and say, hey, would you like some fetal gene therapy? Because they might not be very happy if you did that. So we have to work with cells, and then we have to work with animals, mice usually. I've done the same thing with sheep, and it went on like that. And we learned through various techniques of how to inject into a fetal mouse, how to put viruses in different regions in the mouse and test what we were doing, and seeing if we could get it to work. But one particular route that we got very, very good at was injecting, so this is a mouse fetus here. In mice, they're like string, like beads of different, lots of embryos in one mother, and this is just one of them. And we can inject what we call yolk sac vessels. In other words, these vessels will take your virus into the blood of the animal, so you're delivering to the whole circulation. And we found that, this particular virus that we use makes cells go blue. Doesn't matter how, they're just expressing a gene inside, and they've got their own way of reporting whether they're working or not. So you can see these pictures of the liver over up to a whole year, that we had done these injections and the liver was expressing genes for a whole year. So we're proving we could deliver the genes to the mouse circulation by doing these injections and getting permanent expression of a gene. That was good news. That means we might be able to do it. We then used a different type of virus, and that was the adenovirus, because it's so powerful at infecting cells and expressing genes. So we did this, and there's two graphs. It doesn't really matter, but if you look at the bottom graph, that's an injection into an adult, and in a very short 2 weeks period, we were injecting a virus carrying this factor IX gene because we were very interested in this haemophilia idea, curing haemophilia. And you could see within 2 weeks that the expression, which is the red line, the bottom graph, red line, it drops immediately, because in an adult, there's an immune system and it just gets rid of the virus, it gets rid of the expression and everything, it just stops it working. But if we did it in a fetus, in a mouse, which is the graph above, it takes a lot longer to remove the gene expression by the virus, and also even if we inject more of it, either as protein or as virus, we don't get antibodies against it, but in an adult we do. So we've proven that if you deliver genes before birth, the immune system doesn't realise it, and it lets us get away with it. So we're getting the gene in. So that was exciting too. So this virus isn't the right one to use because it doesn't last very long. It gets into cells and then the cells get rid of it. So it's not good. That's no good for long-term gene therapy. So what we did was we got all these. Don't worry about this; it'll be complicated. These are viruses, but the DNA inside the virus, the genome of the virus, carrying the gene we want to deliver. So each of these lines is representing a different virus we used, and ultimately all you need to know is that we wanted to embark on delivering the factor IX gene to mice. And the mice that we were going to choose to do this were genetically engineered that they were haemophiliac mice. They had haemophilia. So if you cut one of these mice, it would carry on bleeding. And if we could deliver the gene effectively using that injection methodology, could we cure that mouse? One thing I didn't tell you is the reason why we use these viruses is because they cut the genome open of a cell and they insert themselves. That's what HIV does. And it's become permanently in the chromosome of the person. That's why you can't get rid of HIV once you've caught it. It's in your genome. And these viruses like to go into what we call places in the genome which are open. So they cut the genome, they insert themselves, these viruses, and that's called integration. They become part of the genome of the host. And then we did these experiments. On the top left-hand side there, we took a load of haemophiliac mice, we injected them fetally, and we cured them. And we published it. It was the first known permanent cure of haemophilia in a mouse model. It was wonderful. Fetal gene therapy was working. We were so excited that we thought, right, everyone's going to say this is good, we can do this now, patients can be treated full birth, and it's all very, very exciting. But we used a different virus, which is the graph below here, and suddenly these levels, these lines going up, is measurement of the protein, the factor IX being produced by the virus in the cells. And for the top one, you can see that it goes up and down, the levels of factor IX go up and down, which it does in all of us. Our levels of factor IX go up and down. But in the bottom graph, the levels of factor IX started going up and up and up. And we found that this was associated, these levels of factor IX increasing, basically we'd cause tumours in every mouse we chose, virtually every mouse. So we thought we'd cured fetuses for the mice for haemophilia, and all we did was one type of virus was fine, another type of virus was actually causing tumours in the mice. Disaster. This is what tumours look like. You can see a normal liver, next to it this bulbous, this is a tumour, there's tumours in the lung here, tumours, just a nightmare, right. So, and we could look at the cells individually, and we do that in the microscope, and we stain them, and we look and see what they look like, and they did in fact look like tumour cells. So that we knew that too. We then did some clever molecular techniques which asked the question, when the virus goes into the genome of the cell, where does it go? What caused those tumours? And we found that the virus was integrating all over the genome, but we found by doing these clever techniques what genes in the mouse genome, what genes have been inserted into by the virus. And were they changed in expression? And the answer was these were pre-cancer genes. We turned these cancer genes into real cancer genes, became uncontrollably expressed and causing tumours in the mice. So we have a virus here which could possibly cause what we call oncogenesis or tumour formation in mice, and therefore you can't now go into a patient and do that because you can't cure a child 1 minute and next minute, 10 years later they get tumours. We don't want that. And that's already been found in some of these other clean gene therapy trials, as I mentioned to you. So we've got this problem that we can do gene therapy, but we can't do it so it doesn't harm the host. So we also then turned this on its head and found out within the mice we can discover cancer genes by mutating them with the virus and identifying they go out of control and they're cancer genes. So we're discovering new genes which are causing liver cancer in these mice. So that's a good thing, but you don't want to do it in humans. And we put all these genes together in networks and we ask whether or not they are cancer networks. And we did this is called bioinformatics. And we identify that the gene networks which are changed in expression because of the virus causing mutations are actually cancer networks. So it's kind of valuable for us to identify new cancer genes, but not good for the patient, when this case was a mouse. In many trials, and don't worry about all these, it's a bit complicated, in many different trials, the same thing has happened. Individuals have been treated for no immunity, and they've developed tumours, in fact leukemias, or there's been statements or there's been individuals that we found that have been treated by gene therapy that had side effects, and this was because of the virus we used in gene therapy. So, the FDA came along in many of these cases and stopped the trials. The FDA, they're the regulators of what we do. And we have in the UK, it's called the MHRA, and the EMEA are for Europe. So these are the regulators that say whether it's safe or not to do this kind of thing to patients. And the FDA put a stop on so many of these trials, and they said, right, we need a way to test every virus you use for gene therapy. We need a way to test its safety. We need a new model. We need to find out what's going wrong, redesign these viruses, take more bits out of them that could be causing the problem, and then if they're safe, these viruses, then you can talk about doing gene therapy. So I won't go down the list, but their list of what they wanted from a model to test the viruses was quite complicated. Obviously, they should ask this question. They shouldn't just let people do anything they want. And then, in line with this, the MRC, or a section of the MRC called the NC3Rs, these are people that award you money to develop these models to test the virus safety. One of them was called the Crack-It Challenge. And what happened was the Crack-It Challenge asked the same questions as the FDA. It wanted us to be able to evaluate safety in humans, not in mice, but we can't go in humans, so we have to find an alternative which is representative of humans. And we want you to do lots of clever molecular analyses to show that the virus does cause problems and identify what problems the virus have got that's causing the problem, then modify the virus so it doesn't do it. So we won this challenge. It was across Europe, and we were one of the 3 groups that won the challenge, and then between the 3 groups we had to compete with the other 2 groups. And I remember going into Wellcome Trust building to give my talk about whether we could compete with the other 2 groups, and the other 2 groups had joined together against our group, and I thought, well, there's no hope in hell we're going to win this grant, but for some stupid reason they gave it to us, and we won the grant, and then we did all the research. And so what we did was we developed a model where we take, as Christina just mentioned to you, we took stem cells, or cells that, ordinary cells, human cells, which can be converted back into stem cells, which can then be changed into liver cells. And then we can infect those cells with the viruses and see if the liver cells proliferate, in other words, become cancerous, or whether the viruses have these bad effects on these cells. So it's all a bit higgledy-piggledy and complex, but we developed this platform, and because we were using human cells, we were able to say, well, our model tests the virus in a human cell background rather than a mouse. Because what's been generally perceived these days is if you use mice for experiments, first, I hate using animals for experiments. That's why I wanted to get away from that, and that's why I did this. But also, mice don't always represent accurately the human situation. They only live a couple of years. We live a lot longer than that. And so, what happens in our cells and our bodies is going to be a bit different to mice. And if mice are getting tumours, it doesn't mean a human's going to get a tumour. So we can't always trust the mouse experiments when you're going into the clinic with a treatment. So we run that, and we first of all developed a assay where if a person's going to be treated for gene therapy and you're going to get their genome cut open, we have to know that that person is able to repair that. It's the same when you have chemotherapy or radiotherapy. The cells in the body are irradiated or treated with chemicals, the cells, the tumour cells, and the normal cells get it too. But if the normal cells can repair themselves, then they will stay alive, and the tumour cells won't because they've been directly hit by the radiation. So we need to know whether a patient who's going to be treated by gene therapy can repair the DNA back together again. So we did that, and we have this assay where we can see, after 5 hours, we can see lots of dots, which means we've treated these cells with the radiation or gene therapy, and there's lots of breaks of their DNA. And then after 24 hours, if a person's competent to repair their DNA, all those dots go away. Okay? Simple as that. Underneath is a cell line that can't repair its DNA, so these dots remain. Now, you don't want to go and get treated for chemotherapy if you had cancer when you've got a problem and you can't repair your DNA. It will kill you. You'll be stone dead. And that's what has happened to a number of individuals who've been treated with radiotherapy. Then we didn't know that they can't repair their own DNA. So we've developed this test that we can test every patient that can either be treated for cancer or gene therapy. We can test whether they can tolerate the treatment. And that isn't available at the moment. You can go to the hospital, they won't know, but we can tell them that. So we're developing that. And there's some typical sort of graphs of patient T cells where after 5 hours there's damage, and then after 24 hours that damage, don't worry about that, the graph is saying. It's just simply telling us that there is damage and it's repaired. And that blue outlier, that line there, is the cell line that can't repair. We want patient T cells to all look like all the other colours, not the blue line. So we have this assay. We then developed this stem cell assay where we would infect the cells. We would actually turn the stem cells into liver cells. We would infect the cells with the viruses, and we'd do a whole series of tests on where did the virus go into the cells. Did they hit cancer genes? Do they change the expression of cancer genes? Do they change the type of cell it is? Is the cell... it's a methylome thing. Don't worry. It's a factor that you don't want to alter if you don't want cancer. That's all. So we tested a lot of sort of complex molecular assays to make sure, or to find out whether a virus was causing bad effects, side effects on cells. And if we can identify that for our particular virus and modify our virus, then put it back into the system, and it doesn't do anything bad, we've found the right virus. So that's what we've developed. So we took stem cells. These cells can be, once you've made a stem cell, you can actually turn them into any type of tissue you like. So we grow them into little liver organoids. We convert stem cells into livers, into little round livers, and we can treat them with our viruses. So we're kind of mimicking a human liver in a test tube. Right, so we have these cells, we make them into liver-type cells, we then infect them with the viruses, and sorry it's about the colours, but we subject 1, 2, 3, 4, 5 different tests to the cells to see if the virus has caused a side effect. And that now we're making available to test gene therapy viruses. So now we would say if a company wants to go and treat an individual with a gene therapy virus, they come to us first. We test the virus to make sure it's safe. We tell them how to modify it if it's not, and then we will give them that information back. We're providing a service. 1, can the patient repair their own DNA? 2, is the virus safe? And that's what we're offering. This is what we developed. Complicated stuff. Those green round circles at the end there are infected liver organoid cells, and they're green because the virus, we used a virus that makes them green, so we knew we were getting the virus to infect the cells, and we knew how much infection we were getting. We had to prove all that because we had to show that we are infecting the cells before we can test whether the virus had a side effect. So we took cells, we characterized them, we turned them into organoids, and then we look at whether or not there are side effects. So we set up this protocol here. We called it HinGetox. We take the stem cells, we just subject them to a whole load of tests for the virus to see if the virus is causing a side effect. And then for each of these things here, running up this horrid, terrible background I've used on this slide, we can tell for each virus, one is called AAV ApoE, another one's called PHV. These are just different viruses. We can put them on a scale as to how safe they are. And we found that, for instance, PHV is the most unsafe, and AAV ApoE happens to be the safest one. So we're able to find a virus by our screening technology if it's safe. So overall, the future is we've got a system here that we can do this, and don't worry about this slide. We did some market research. We founded a company here at Brunel called Testavec Limited, and we did some market research to see if we have any competitors, and we found there wasn't any competitors. We've had lots of meetings with the FDA. We put IP on our technology so we can own it and make it available to others to use. And interestingly, future developments, and you may have seen this just in a couple of days ago on the news, this young girl who had all the different treatments. She had a type of leukemia, and she had all sorts of different types of treatments that didn't work. And she had this new technology called CAR-T, which means you take cells, you change them so they can be put back into the patient and attack the patient's tumours and kill the tumours. It's the latest technology now. You may have heard of it. CAR-T it's like what everyone's always wanted. Take your own body's defense system, boost it, put it back in, and kill the tumor if you've got one. That's where it's going at the moment. And this poor young girl came to Great Ormond Street with no help at all and went through a whole different treatments. And the CAR-T therapy, which is basically boosting her own immune system, was used, and it's worked. And she's now, it seems clear. And she's been interviewed and everything else. So what we then were worried about is these CAR-T cells, they are the patient's immune system, which is modified by putting genes in them to make them more aggressive against tumours when they're put in. But the way you do that is by, again, putting a gene therapy vector into that patient's cells. Again, there we go all round again. Oh gosh, are we going to cause a side effect? So we need our system to test. And so we've just won another grant, which is called the Crack It T-Alert grant, where we're developing our model for this technology to make sure it's safe. So I've tried to sort of rush through it and give you an overview of what we do, but it's all very meaningful, and we have a couple of products now for Testavec. One is called DNA Rep, which is available to test people's ability to be treated for cancer, and one is called HinGetox, which, as I mentioned, is testing side effects of vectors and making sure that there aren't any, or modifying the viruses so that they are safe. And I have to say, lots of people involved in this, including Great Ormond Street, Novartis, GSK sponsors, and so on. All these wonderful people. It's not me, it's all these wonderful scientists that got us to this point. And that's kind of, I think, yes, that's me finished. Thank you.